All right, welcome to our first remote lecture for the rest of the semester on poxviruses. So poxviruses are unique in that they're very, very large DNA viruses that can cause a lot of damage. They're DNA viruses, they're large, and they replicate entirely in the cytoplasm. They do not go to the nucleus. And the most famous pox virus is smallpox. Smallpox is caused by the variola virus. There are a couple different strains of variola virus that we'll talk about. And smallpox has killed more people than pretty much anything else in the history of humanity. In the 20th century, smallpox has killed 500 million people, which is just a staggering number of people. And smallpox, we think, has been around for a very, very long time. And we have seen a lot of evidence of smallpox throughout history, including mummified remains of people that we can see they had pox lesions on. Smallpox has been used as a bioweapon before, most notably by colonialists, and they came to the New World and they intentionally spread smallpox to the native populations. So what does smallpox look like? Well, like I mentioned before, there are two strains. So you've got the major strain and you've got the minor strain. The major strain is overall, we say overall, about a 30% mortality rate. So very, very serious. And the ordinary smallpox is what we're going to talk about primarily. And this is most cases, right? This is, 85–90% of cases. Then you've got some cases that are more extreme, the malignant smallpox, which has a much, much higher mortality rate, hemorrhagic smallpox. So this is when you get these lesions, but internally all over your body as well, which is also extremely fatal. And then the last type of smallpox of the variola major is the vaccine modified. And so this is when somebody gets smallpox after they've had the vaccine, and it's not as bad. So after somebody gets vaccinated, occasionally the vaccine isn't 100% effective. They can still get smallpox, but it's a much, much more mild disease. And then we've got variola minor. Variola minor is a milder disease, and this is why it's the minor instead versus the major. So the minor virus, though, is still pretty fatal, 1–2%, and that's really, if we think about our current pandemic, we think it has an overall fatality rate of 2-ish%, 2–3%, and look at how much disruption this has caused worldwide. So variola minor, not nearly as bad as variola major, but still not a fun thing to get. Okay, clinical presentation of smallpox. This is honestly one of the more horrific diseases. The incubation period is quite long. 12 to 14 days, and luckily you're not contagious during the incubation period. Then when your first symptoms appear, pretty good fever, a very bad headache, backache, sometimes you get vomiting and diarrhea. Delirium is also a relatively common symptom. Now, this lasts 2 to 4 days, and you're relatively contagious. So this would be like day 1 of symptoms when it starts. but we're really at, say, day 14 if you include the incubation period. But we're going to start now on day 1 of symptoms. And so this fever, headache, and so forth, this is going to last 2 to 4 days. Now, after the fever breaks, so about day 3, 4, then you get a rash, and it's on your tongue and in your mouth. And when you get a rash on a mucous membrane, it's called an anthem. This appears on roughly day 4, and then it becomes blistery. And when those blisters erupt on about the next day, so day 5-ish, then you're very, very, very contagious, and you get a huge number of virus particles that are going to occur in your saliva. You can also imagine it's difficult to eat and drink during this period of time. And these lesions can also appear in your upper respiratory tract. And when you cough or spit or talk, you are releasing large amounts of these viral and infectious virions. And the R0 value is about 3. Now, remember, this is a kind of a statistical average. It depends on a lot of things. In theory, this should be higher, and I think the reason it's we're about 3 here is because when you're experiencing smallpox, you don't want to get out of bed. You are miserable, you are really, really sick, you're not really out there exploring and coming in contact with a lot of other people. You're infecting your close friends, your family, your healthcare workers here. Now, you've got these blisters that are in your mouth and in your trachea and on your tongue, which is exceedingly uncomfortable. And then after the rash in the mouth starts to blister over, then you get the blisters on your skin. So first, a red rash appears on your skin, and when you get a rash on the outside, of your skin, we call that an exanthem. It starts off as a red rash, and then it becomes vesicular, and then those vesicles, break and scab over. Now, really importantly, the rash, the order it appears. So you get it on your face, and you get it on your extremities. So that would be your hands and arms, your legs, and your feet. And you get fewer blisters, fewer marks on the center of your body. So this is called a centrifugal rash. If you think about spinning something in a centrifuge, everything goes to the outside. So you get more of the pox on the periphery of your body. It's not completely exclusive, but you get more on the outside. And this is important for diagnosis. In the second week of symptoms, so this would be day 14 of symptoms, the rash starts to crust over on your skin. And then after an additional week, so where it would be like at day 21, in your third week of symptoms, the scabs start to fall off. So in summary, you've got a long incubation period where you're not contagious. Then you're in the prodromal phase where you have a fever, headache, last a couple days. Then the fever stops, and then you get the rash to appear first in your mouth, in your upper respiratory tract, and then on the outside of your body. And these blisters are really, really painful, and you're really miserable, and you don't want to move around outside at all. If we take a look at the rash, this is the centrifugal pattern. So you've got more pox marks on the extremities than you do in the center. There's still a couple on your trunk, but less. This is in contrast to, say, chickenpox, which is a very different virus. In chickenpox, you get more bumps on your trunk and you get fewer on your extremities. And so different rashes that we're going to talk about throughout the semester, they appear in different orders or in different places in your body, and that is important for diagnosis. And then here you can see is a rash on a child, and he still has a few bumps on his stomach, but he's got a lot more on his face and on his legs. So this is smallpox, and it has killed millions and millions and millions and millions and millions and millions of people since there have been people. This has been one of the biggest scourges of humanity, with a very, very high mortality rate. This actually used to be so common that if everybody would get this, there'd be periodic outbreaks, epidemics, and everybody would get infected, and 30% of the people would die, and then the people that survived would be disfigured. And so even after this crossed over and the pox go away, you're still disfigured. And it was actually considered really rare to not be disfigured from smallpox. Like, that's how common this was, that if you didn't have a disfigurement, then that was something notable. But let's look at some other pox viruses. There are pox viruses that infect insects, and there are ones that infect different types of animals. Many pox viruses are zoonotic. They can infect different types of animals, but some are localized to just one host. And we're very lucky that smallpox only infects people, right? So right here, variola major and variola minor, this is smallpox, only infects people. And this is the only human disease to be completely eradicated. There are no more naturally occurring cases of smallpox, thanks to a wonderful vaccination campaign. There are some other pox viruses, though. So if we look at other pox viruses, we've got monkeypox, and this can infect several different animals, including people. This is an example of monkeypox. The sorts of blisters you get. The case fatality rate can vary a little bit. People usually get this by coming in contact with a reservoir like prairie dogs, and this has been found in America. This was transported to America. It's also in Africa. Another one of concern is molluscum contagiosum, which is a fun thing to say. It sounds like something in Harry Potter. Molluscum contagiosum! And that is shown here. This is something that is spread through direct contact. It's only in people. And this, you get these pox lesions. They often have a dimple in the middle of them. Through direct contact, could be sexual contact, doesn't have to be sexual contact, but direct contact. And this is a bigger problem for people who are immunocompromised. And one of the concerns with this in immunocompromised individuals is that even though this is usually self-limiting, so the virus gets taken out by your immune system or by medication, and one of the problems is these blisters, these sores actually stay for a long time. It can be weeks or months before they completely resolve, and they can be an entry point for secondary bacterial infections. Secondary bacterial infections, and that's a particular problem if you're already immunocompromised. These other pox viruses, ORF is something that's in livestock. It can infect people if you're immunocompromised and if you're in contact with a reservoir. Vaccinia virus, right? So this is in cows, and this is our vaccine. Of note, vaccinia virus also protects against, or an immunization with vaccinia virus protects against smallpox and monkeypox. And there's a few other pox viruses, but we're going to focus on just these. Now, almost all of our information about poxvirus biology really comes from studying the things like a cowpox or a vaccinia, because we eradicated smallpox in the 70s, and so a lot of the newer techniques and a lot of the newer scientific methods we didn't have before smallpox was eradicated. So almost everything we know about the biology and the genome of this family has come from studying things like vaccinia. Now, of note, these viruses are very large. They can be up to, say, like 300 micrometers. Nanometers. Sorry, up to 300 nanometers. They're really, really big for a virus. This is bigger than some tiny bacteria. So they have a very large size. They also have a very large double-stranded DNA genome. And in the genome, you've got a linear genome. The ends of the genome are inverted terminal repeats, which they actually kind of loop a little bit on top of themselves. And then you've got your essential genes that are in the center of the genome and non-essential genes that are kind of on the exterior. There is a really cool paper that I've put into the module that has some investigations into how the genome of poxviruses can adapt to different situations. And there's also a fun little video that the authors of that paper did, kind of explaining their paper, which is also uploaded in your Canvas module, so that's kind of a fun thing. Now, poxvirus structure. This is a complex morphology, meaning it doesn't fit neatly into our other categories. So here's the cartoon, here's the electron micrograph. Notice the barbell shape. That doesn't show up very well, right there, barbell shape of the core. And then you've got these lateral bodies over here, which contain some miscellaneous enzymes. But the genome and so forth, the essential things are going to be in this core. And then there's some useful enzymes that are stored in these lateral bodies. And then we've got multiple membranes. So you can see the membrane here in the electron micrograph. Over here, we've got a core envelope right here. And we also have extra membranes. And there's different versions of these particles that can have different numbers of membranes. We'll talk about that next. Okay, so we've got our mature virion and our enveloped virion. The mature virion is abbreviated here, MV. Your enveloped virion is abbreviated here, EV. Now, in your textbook or in different sources, they might have different ways of calling these, and we'll get to address that why. But if you understand the principles of what we're talking about, then you should be able to follow along no matter what source you're in. And we'll talk about how these extra membranes occur. So the take-home point for just this moment is that you can have different numbers of these external membranes here. Now, this does mean that when you go through the replication cycle, you have to have an extra step. If you've got an extra membrane, you've got to have an extra uncoding step. So the enveloped virion, which is pictured here, has that extra envelope. So once it enters, it goes into the endosome, and the outer membrane gets disrupted, and then the inner membrane fuses with the endosome to release the core into the cytosol. Now, if you only have one of those external membranes in the mature virion, then that enters, it goes to the endosome, and then in the low pH environment, that viral envelope, that viral membrane fuses with the endosome and then releases the core into the cytoplasm or the cytosol. And then from there, both of the replication cycles are going to be the same. And so we'll walk through the replication cycle, and then you can see where they pick up their extra membranes. Okay. So the number one really important thing to remember about poxvirus replication is all poxviruses, this is a hallmark of poxvirus replication, they all replicate entirely in the cytosol, in the cytoplasm, so they do not enter the nucleus. Poxviruses, because they don't go into the nucleus, that means they have to bring all of their enzymes in with them. So that means they're going to have to bring in their own DNA polymerase. They're going to have to bring in their own RNA polymerase. Oops. They have to bring in their, oh, I got this written twice, don't I? There was a copy-paste error there. They have to bring in their own DNA polymerase. They have to bring in their own RNA polymerase. They have to bring in their own transcription factors. They have to cap their mRNAs. Because remember, the ribosome will not start translation on an uncapped mRNA. This is a control mechanism in the cell. So if you're not going to go through the nucleus and get capped, then you've got to be able to put the 5' methyl G cap on by yourself. And they also have their own poly-A polymerase, so they can add the poly-A tail. So because poxviruses are replicating entirely in the cytoplasm and not the nucleus, they have to bring enzymes to do all of these things. So poxviruses, again, very big viruses, very complex transcriptional regulation. And so they regulate their genes in 3 stages. We've got the early stage, which they're going to need right away for gene replication and so forth, and evading the immune system. And then at the end of the early stage, they make the transcription factors for the intermediate phase. And then the intermediate phase is going to make the transcription factors for the late phase. And so this sets up like a timer effect, or like a really cool Rube Goldberg device, right? So first you got to do the early, then that triggers the intermediate, then that triggers the late. And this ensures that things get made in the right order, and you're not wasting energy making the late genes, at the wrong time. And so this ensures a very orderly viral replication cycle. Okay, so what do we need to do if we're a poxvirus? So first we have to enter through the cell, right? We come in through first, actually, right? We have to bind first, attach, and then enter. So we're going to bind to sugars on the surface of the cell, and these would be glycosaminoglycans, or GAGs. Examples could be chondroitin, or heparin. So we're going to bind to sugars on the outside of the cell, and these are very widely expressed and allow the virus to have a broad cellular tropism. So first we bind, and then we enter. So now we're going to enter, and in the endosome, the pH is going to lower, and that's going to trigger viral fusion. So the envelope of the virus merges with the envelope of the endosome, or the envelope of the virus merges with the membrane of the endosome. So we've got membrane fusion. 2 membranes fuse, and that releases the core into the cytoplasm. Now, this also opens up the core, and remember those lateral bodies that had extra enzymes? They can start interfering with the immune system. Now, right after the core is released, we're going to start our early mRNAs. So we're going to get the things we need for DNA replication started. We're going to create some more interference with the immune system so we can continue to replicate uninterrupted. And then we're going to start transcribing the proteins that you need for the next stage, for the intermediate mRNA. And then that's going to go through and it's going to trigger the late mRNA expression. And this is going to include things that you need to package into the new virions, and it's going to include structural proteins for making the capsid and so forth. So after you've got all of your parts, right, after you've got all the things that you need, now you need to start assembling. And assembly involves massive rearrangement of the cytoskeleton, also involves massive rearrangement of the Golgi. And this is really kind of destroying your cells. You get these viral factories where you really reorganize the cytoplasm, the cytoskeleton, you're stealing membranes from the Golgi to make the new viral particles. You're also stealing membranes from the ER as well. So you make more genomes, right? You got more DNA, you're going to have all of the proteins that you need, your structural proteins and so forth, and you get an early membrane. It reorganizes the membranes in the endomembrane system, and they form these crescent shapes, and then these early membranes, they start to form around the genomes and the required proteins, and you wind up with this mature particle. Now, this particle is still infectious, but it's not done yet. Now, you wrap this particle. You wrap the particle in membranes derived from the Golgi. Now, at this point, you actually have 3 membranes. You've got the core membrane, and then you've got another membrane around here, the mature membrane, and now you've got this Golgi membrane, right? So now you've got 3 envelopes. You've stolen lipids, you've stolen membranes from the ER and from the Golgi, and so now you've got this wrapped particle here. Now, this is going to completely reorganize the cytoskeleton. It's going to use microtubules to push it to the plasma membrane, and there the fusion is going to occur between this wrapper, this Golgi wrapper. The Golgi wrapper merges or fuses with the plasma membrane, and then the cell ejects the virus, or the virus leaves the cell. And so you lose that Golgi wrapper when you leave the cell. But you start replicating, and eventually the cell just dies. It can't take any more. And so you've got your mature virions. I'm going to, right, so I'm going to make some more annotations here. All right, so these mature virions, they leave the cell, they're ready to go on and infect other cells. Okay. But when the cell lyses, you've still got these wrapped viruses in place that have this extra 3rd membrane. These are still infectious. And this is that extra membrane that we saw on the previous slide, right here. So that's where we get this extra membrane from. So this is just they haven't been released from the cell yet, but they're still infectious. So when the cell dies, they just get released. So you've got these two infectious particles. Now, moving on. Oh, I should say, let's pause here and let's just highlight. This. That's not a highlighter. That's an eraser. All right, there we go. Immune modulation. This is really, really important. So in the lateral bodies, you've got some enzymes and proteins that can start evading the immune system. And during early gene expression, you're going to turn on some of these immunomodulatory proteins. And pox viruses really do a number on your immune system that allows them to cause very, very serious infection. So we're gonna talk about those next. So we've got viroceptors. We've got viroceptors and we've got virokines, and this is kind of a play on words. This would be virus receptors. So virus receptors and virus cytokines. So these proteins are going to vary by each virus. So all pox viruses are going to have a slightly different immune evasion program, but there's a few things that are similar. One is pretty much all pox viruses have some way of interfering with the antiviral state. So you guys need to remember from the previous material, the antiviral state. We're going to be talking about that pretty much all semester. So viroceptors are viral-produced receptors, or they're altered cellular receptors that aren't anchored in the transmembrane anymore. And if they're not anchored, then that means they are secreted. And these receptors are things that bind interferon. So we've got viral mimics of interferon alpha beta and interferon gamma receptors. So that means these proteins are designed to bind interferon, but they're secreted. So instead of interferon binding to the surface of the cell and then starting the antiviral state, which would inhibit pox virus replication, these viroreceptors are secreted. They bind up interferon, and then interferon can't do its job. Interferon can't get where it needs to be. Interferon can't bind to a cell and tell it to shut down the ribosomes and so forth. So this is very, very effective at antagonizing interferon, binding interferon, and helping the virus avoid the innate immune system. Another strategy is they interfere with interleukin 18. So IL stands for interleukin. And interleukin 18 is going to stimulate interferon. So this indirectly stops interferon. So if we stop interleukin, we stop interferon. If we stop interleukin, the end result really is that we are really hosing up the helper T cell responses as well, and we're going to hose up cytotoxic T cells as well. So this really cripples the antiviral immune response. But they're not done. They also can modulate different chemokines, so they can regulate cell behavior. They can prevent leukocytes from getting activated, and they can directly interfere with complement. And we have this complement control protein. It's the vaccinia complement control protein, which they cut out a couple letters and they abbreviate that VCP. VCP leads to the decay of C3 convertase. And if you guys remember from the complement system, C3 convertase is that big domino. Once you push over C3, then the whole complement system is triggered. So they prevent that. They break down C3 convertase. And this is just a sampling of different strategies that pox viruses can do to evade the immune system. It's going to vary a little bit with each different pox virus, but in summary, pox viruses really, really cripple your antiviral immune response. You should focus on the ones that are listed on this PowerPoint slide. They talk about more in your book. And if you just look on PubMed for pox virus and immune evasion, you'll see lots more different mechanisms that different pox viruses can use. So pox viruses have been infecting us for thousands of years. So there have been some coevolutionary events happening where people have developed some resistance to smallpox. And they found that different people have different susceptibilities to smallpox. So they found that actually by blood type, if you are type A, you are much more likely to get smallpox, more likely to have it be severe, and more likely to die of smallpox. So that's not good luck for me. Also, and this is related but different, so we're talking about who's more susceptible. Here we're talking about resistance. So they found that people that have a deletion in CCR5 here at this residue have resistance to smallpox, and that also confers resistance to HIV, which is pretty handy. Okay, smallpox was eradicated, so yay! Smallpox is the only human disease that has been completely eradicated. And there's a few reasons why this was possible, okay? So the biggest one is that smallpox only infects people. If this infected other animals, it would be much, much harder to control. You'd have to either vaccinate them or control the other animals, right? Because this only infects people. That made this possible. Also, we were able to make a vaccine. And because this has pretty obvious symptoms, it was, relatively speaking, easy to surveil for it and find people who were infected. And so this was a 10-year campaign that the World Health Organization undertook, and they tried to vaccinate everyone they could find. And then they went around searching in remote villages for years, searching for any trace of smallpox infection so they could make sure they vaccinated everybody and got everybody treated if that was an option. And to the point where they were offering money to not just the healthcare worker, but also the patient. If you have smallpox, let us know, we'll give you money and a vaccine. And so after a 10-year program, they were able to eradicate smallpox. So there are no naturally occurring cases of smallpox anymore. There are some samples in a couple laboratories, and there have been some laboratory incidences, but there's been no natural cases of smallpox. Now, because this disease has been such a blight on humanity, lots of people have tried to find ways to prevent it. So people have tried vaccination or immunization in relatively unsafe ways for at least hundreds of years. So looking back, I lost my cursor. How do I lose a cursor? All right. So looking back, we can see people tried to inoculate others by giving them low doses of smallpox scabs from people who recovered. People doing this, again, normally smallpox is like 30% or more fatal. So this was pretty risky, like 2 or 3% of people died, but that was better than 30%. People were kind of desperate. And then we've talked about Jenner before, and he used cowpox, and this was much, much better. This was much safer than the previous method. So he was right, luckily, and he experimented on the child of an employee, which we've talked about before is not ideal, not true consent, but it worked. They were very lucky. And then we used cowpox for a really long time. In the 1800s, what people did is they would vaccinate one person, and when you vaccinated a person, you get a blister, and then you would take pus from the blister of the vaccinated person, and then you would transfer it to the arm of another person. And this was definitely not safe. And sometimes this got contaminated with actual smallpox if a person also happened to be infected. So this was banned in 1898. Now, keep in mind, people were still pretty desperate because smallpox was a very real threat where, 30% of people died. So then, in the late 1800s, we actually started producing a reliable vaccine. And this is kind of crazy to me because we continued to use this vaccine up until the 1970s. And the way this was produced, this vaccine is called Dryvax. And this was produced in cows, where you took cowpox lesions from one cow and then you infected another calf. And then the lesions that were on the cow, you scraped them off and collected that in a jar. Then you added some glycerin to preserve it and kind of make a pulp, and then antibiotics were added to prevent contamination. And this was used, this was centrifuged down, and then you collected the liquid, and that was the vaccine. And we used that for, about 100 years, up through the 1970s. The World Health Organization eliminated smallpox with this method. Well, now we can do this in tissue culture. And this is safer. You've got less contaminating proteins. This is more pure. We can do this in tissue culture. So it's the same strain of vaccinia, but grown in tissue culture and much more pure and more reliable, and the dose is more consistent. With Dryvax and with the newer version, AKM 2000, the idea is you use a needle and you stick it into the skin of the arm. And you poke the person several times. And the idea is to create a small, localized infection. And this is very, very, very important. You want the body to develop a localized immune response. And this virus can replicate. It's attenuated. Oops. It can replicate, but it's a little dangerous. It's not. We've got about like a good safe vaccine might have an error, an adverse reaction of like 1 in a million. That would be a nice safe vaccine. This here, we've got adverse reactions in about 1,000 out of every million. And a lot of that has to do with, is the person, do they have any contraindications, and do they scratch this, do they scratch the pustule, right? It really needs to stay localized. You just want a small little replication site of infection, a small little focal infection in your arm. But what happens is people scratch this, and then they, say scratch their eye, or they rub someplace else on their face, and then you can get a systemic infection, and then that can be much more serious. So the idea is they take the vaccine, they create just a small little localized thing in the skin here, and then you get a local infection, and then that scabs over, and then you're immunized. And this is still the most effective vaccine. It works the fastest, but again, it's not quite as safe as we like modern vaccines to be. This 1,000 in a million is a little problematic, especially if you have contraindications. So we'll talk about that next. All right, so if you are immunocompromised, you cannot get this vaccine because it's attenuated. That means it's still living and it's still replicating, and it can go systemic and it can be fatal. You also cannot get this vaccine if you have eczema or atopic dermatitis, because then it can spread all over your body, it spreads systemically, and it can be fatal. So this vaccine is also dangerous if you have some other pre-existing conditions that basically enable this to spread beyond the site of infection. Now, we are very, very lucky that now they have developed a new vaccine. This is attenuated. It's not replicating. Okay, it's called Imvamune. It also has a couple different names depending on the company that makes it. There's a couple brand names for it, but Imvamune. This is the vaccine I would get because I have a history of eczema, so if I took this vaccine, I might die. So Imvamune is the way I would go. It's much, much, much safer. You can give this one to people who are immunocompromised. You can give this one to people who have eczema or other dermatological issues. The gotcha, though, is it takes a little longer. You need 2 shots, and you're not considered immunized or protected until 2 weeks after the booster. So this one, again, much safer, but it takes a little bit longer for you to have the same protection. There are a few treatments for smallpox. We're not, again, a lot of these studies have been done not with smallpox, because smallpox has been eradicated now, but with other pox viruses. So we're not really 100% about a lot of these, but they're better than nothing. We've got a couple drugs here that target the viral polymerase. We've got some others that target different viral enzymes. So if you're not vaccinated, we do have some antivirals that have either been produced or under investigation. So this is the lecture on smallpox. Your book talks about the likelihood of smallpox being used as a bioweapon. My take on that is, yeah, it's theoretically possible it could happen. We don't vaccinate people against smallpox anymore because it's been eradicated, so the population is naive now. We're not immunized to it. Everything is a cost-benefit analysis. Even with the newer vaccine, if the risk of an adverse event to the vaccine, we'll say, is 1 in a million, but if you vaccinate 100 million people, you still have 100 adverse reactions. Is that worth it if the disease has been eradicated? And so public health officials have said, no, there's no reason to vaccinate people if there's no way you're going to get infected. And let's protect that 100 people and 100 million. So that's the public health stance. The only people who get vaccinated now are those who are considered high risk, those who are maybe working out in the lab, or some military personnel get vaccinated. But they have stockpiled the vaccines in case a bioterrorist attack happens. Is it theoretically possible? Yes. Honestly, I'm much more concerned about influenza, and I'm more concerned about, coronavirus, for example, right? So it's still interesting to read in your book. You can read through the possibility people have investigated for destroying all cases of smallpox virus, or should we continue to do research on it, and what is the likelihood of a virus terrorist attack. So that's fun to read, but not, in my opinion, the biggest threat that we're facing. Anyway, this concludes the smallpox lecture. I will get more lectures uploaded for you guys as soon as possible. It's been a crazy week for you guys, I'm sure. It's been a crazy week for me. And though we'll get through this one way or another, we don't have a choice. We'll get through this till the end of the semester. So I hope you guys are doing well, and I will update with more content soon.